In this video, we will be covering chapters 21 and 22, which are about the lymphatic system and how immunity works in our human body. The thing always to remember is that even pathogens have similar needs as our normal cells. That is to say, if it's good for our normal cells, then it's certainly good for pathogens as well. So we have to actively suppress the pathogens that could take over if not unmitigated. That includes fungi, bacteria, viruses. Protists are like eukaryotes. They have a cell nucleus, but they're kind of an in-between. If you think of Giardia, that's a protist pathogen. Parasitic worms, insects, and even cancer cells can be considered a type of pathogenic organism in some regards. And we're always fighting these off. Some of these things actually help us. We do have bacteria and fungi that we call commensal but we do keep them under control. That is, for instance, the bacteria in your mouth actually are beneficial until they get too populous, they get too many of them. And that's part of why brushing your teeth and rinsing your mouth out is important. In any case, cells and pathogens, because they are all cellular in some degree, like viruses are not cells, but they do take over cells. When they do, or when any of these things show up, they have on the surface of their cell membranes what we call antigens. Antigens are molecules that can trigger immune responses. They can maybe create one or several different antibodies, or again, inflammatory type responses. Antigens can be very reactive, that your body spots easily, or sometimes they need a little bit of help. In that case, we have what are called haptens. This is something that may not have antigenicity on its own, but once it's attached to something else, then it becomes antigenic. And we need to look no further than a slide like this. SARS-CoV-2, also known as COVID-19, very, very serious. We lost over 1 million people in the United States alone to this virus, which was what we call a novel virus. The humans hadn't really seen a virus like this before, and so there were varying degrees of ability to respond to it. So the immune system cannot be understated in terms of its importance. And here we have a list of the immune cells. Some of these we've seen before: neutrophils, basophils, eosinophils, macrophages, and monocytes. We've seen these before, lymphocytes. But we are going to add a few more to this list. One is called a B cell, which is you see APC under it. That stands for antigen presenting cell. We have the T cell, and there are more than one kind of T cells out there, but what they do is they are involved in coordinating cell-mediated immune responses. We have a natural killer cell, which is really cool. This is a cell that will attack virus-infected cells and cancer cells. It will find cells that are not expressing the appropriate molecules on the cell surface and kill them. It's really, really cool. Great promise in immunotherapy for cancer. Then there's this fun one over here on the left called a mast cell. It's really closely related to a basophil. But a mast cell is kind of a first alert cell. It's one that it tends to be a little more ready to respond. It's one of the first responders in an immune, particularly an inflammatory immune situation. And then lastly, we have this dendritic cell. So we have B cells that are antigen presenting cells, dendritic cells that are antigen presenting cells, and macrophages can also be antigen presenting cells. So, several new cells to the list. We saw this figure in blood, but it's a little more complete this time, and that is to say hematopoiesis, going from left the hematopoietic stem cell, which gives rise to any and all of these cells, depending on what signals are given to the cells as they are developing. For instance, we have a myeloid precursor, which gives rise to all of the cells that we were familiar with from blood: neutrophils, basophils, eosinophils, monocytes, and so forth. But we're adding a few cells here. There's a dendritic cell, here's macrophage. And then on the left, this is more lymphocytic in nature. So these run in the lymphatic tissue and lymphatic organs and give rise to things like antibody cells and what we call memory cells. Memory cells are why we immunize. If you are exposed to a particular pathogen, particularly a viral pathogen, you will have cells that remain that are ready to respond quicker than the first time around. And so those are called memory cells. When it comes to our defenses, we have two sort of major categories of immune defenses. And the verbiage can get a little weird here, so I want you to make sure that you understand the concept correctly. The first we have is called non-specific. This includes your skin, this includes antibacterial proteins on secretions, mucin, all sorts of things. It doesn't matter what the pathogen is, those things are going to be there, and you are born with that ability. So now, but you have to be careful here because you do, before you are born, inherit antibodies from your mother. So yes, while you were born with them, you actually weren't really born with them. You were the recipient of them. So that does not count in this. All right, that's the rub. Even though you're born with antibodies from your mother, you didn't in fact make those antibodies yourself. All right, so that's one thing I want to make sure that you're clear on. When we say innate immunity, that means that even if you were never in your mother's womb, you would still have these abilities. All right, they're first and second line, so that includes the barriers and inflammatory responses. Inflammation is considered non-specific. All right. Then we have specific. This is called adaptive or acquired. For instance, you can acquire antibodies from your mother in utero. All right, but those are not innate. All right, those were acquired. You can also gain immunity through exposure. All right, and again, it's specific, so you have to recognize a particular pathogen. It's not just going to happen willy-nilly. It's going to spot an antigen, all right, and respond to that antigen in particular. All right, and it's the third line of defense. So we have the barriers and the chemicals, if you will. Then we have inflammation, which is the second line, and then we have adaptive acquired immunity, which includes mother's antibodies, third. And here we have a nice breakdown of the difference between what we would call innate. And I don't like innate because it implies you're born with it, and I think that gets people confused. But let's call it default immunity, okay? It will happen against any potential foreign invader, and sometimes it'll happen spontaneously or for reasons we don't fully understand. The anatomy of the skin, the secretions, and general structures provide the defense, but it also includes inflammation, which is a chemical and cell-mediated response. It does not have memory, all right? It doesn't create antibodies. On the other hand, adapted immunity is specific for a pathogen. You have cells that recognize this and create antibodies that have, not only are you creating antibodies to meet the infection at that time, but you also do keep track of past infections for a certain period of time, and it depends on the pathogen. As I mentioned a couple of slides back, we have a first line of defense. This is a good explanation of what we see in the first line: skin, mucous membranes, secretions, hairs, tears, saliva, all sorts of things, vomiting, first line of defense, all right? And then we have some chemicals that are also anti-floral, antibacterial. Second line of defense is more cellular and chemical. And I think that brings up a good point, is that while we can categorize these things, the truth of the matter is that they are interwoven with each other. It's a spider web, if you will. But this is still considered part of what we call the innate, the default immunity, if you will, cellular and chemical defenses like antimicrobial proteins. So if a cell gets infected, it will release these cytokines called interferons that will help other cells around that infected cell prevent from being infected. Complement will destroy or elicit inflammation. So fever, which is a manifestation of inflammation, in non-specific white blood cells, macrophages, neutrophils that will phagocytose. And then this is where it sort of connects in. If those pathogens are phagocytosed, a lot of those antigens then get presented to the acquired specific side of immunity. And I did just describe this in the previous slide, how if a virus gets into a cell, that cell will release a cytokine known as interferon, which will help block virus from infecting cells that are responding to that interferon. It's secreted by a virus-infected cell. It diffuses around the cells, triggers inflammation, brings macrophages, natural killer cells, and other things in order to mitigate this viral infection. It is considered that second line because it happens every single time. It's not specific to a given pathogen. So complement is a set of soluble proteins that are made by the liver that have this sort of immunity interfacing with antibody kinds of things. It helped, again, like I said, the default immunity, innate, quote unquote, and the specific cell mediated do work hand in hand. I mean, they're like two dancers on a dance floor. They work together. So what happens in complement, it sort of kind of works hand in hand, but it does happen all the time. Every single infection you have complement. And so what happens is the first thing is that these complement complexes will form and form an attack against a pathogen, particularly bacteria. And then what will happen is that will make that complement bound entity of a target for phagocytes like macrophages, neutrophils, to phagocytose. They'll attach. The complement makes it so that these phagocytes will more easily attach and try to phagocytose these pathogens or pathogen fragments. And thirdly, complement does activate neutrophils and lymphocytes, monocytes, to begin sending out diffusible signals to trigger inflammation in that area. And what does inflammation do? It causes increased blood flow to that area, increased temperature, may signal for platelets and other molecules to arrive and try to fight this infection. Infection is fundamentally a numbers game. How many on offense versus how many on defense? Fever is really, really interesting. We often think of a fever as something that we need to deal with and reduce. And there are times when fever can get excessive. But the actual truth, as we're beginning to understand it, is that fever actually is a good thing. And if that's been known for quite a while. Now, excessive fever is a bad thing. It's just like a little bit of water is good for you, but a lot of water, not so good for you. And the same thing's true with fever. Fever actually elevates the body above 98.6, and it makes pathogens more vulnerable. The increased heat makes pathogens more susceptible to defensive mechanisms. It actually is a more optimal temperature for your body to work. Enzymes, repair cells, and so forth work faster, and you generally have enough nutrients and so forth in order to allow those cells to do their thing. But what fever also does is it makes the pathogens more dependent. They need more nutrients. It actually in some ways sort of contributes to starving the pathogens a little bit because they're burning through due to the increased temperature. A little bit more on fever. Another word for fever is known as pyrexia, an abnormal elevation body temperature. This is actually mediated by the hypothalamus. It's pretty cool that way. The hypothalamus resets the body temperature set point. It is a defensive mechanism. That's because pathogens invade when your body temperature is at or even sometimes slightly below 98.6, which means that's kind of where they want to be. So if you elevate the temperature, now you're taking them out of their comfort zone. So macrophages, when they sense the presence of pathogens and antigens, they will release diffusible substances. They're known as cytokines. We don't use that word here, but they are substances that diffuse through the bloodstream and will tell the hypothalamus to raise the set point of the body temperature. The most common one is known as interleukin 1. Again, it goes to the hypothalamus, and then the hypothalamus will then drive the increase in core body temperature. A little more about inflammation. Now it says inflammation is similar to fever. I tend to blur the lines a little bit because you can have systemic inflammation. So most of the time inflammation will be local, and that's true. So you'll have increased temperature in a local situation. But I do kind of think that they go hand in hand with inflammation, fever, and so forth. In any case, you do get swelling, and we call that tumor. That's because there's increased blood flow, which causes increased leakage of blood plasma out of the capillaries. Part of that increased flow is due to redness because the vasodilation of the capillaries means more blood flow. Certainly heat that will because of more higher blood temperature coming through, vasodilation, pain. That can be due because of the increased pressure, but also release of cytokine intracellular chemicals like prostaglandins, histamine, potassium, so forth. These things all bind pain receptors and trigger pain in those areas. But then, by and large, inflammation is generally beneficial. Triggering initiation of repair, preventing pathogen block, diluting potential pathogenic chemicals, and so forth. But inflammation can be very aggressive as well, and so there's a fine point, and so we might need to mitigate inflammation without completely blocking it. And here is a graphical representation of inflammation, the pillars of inflammation: heat, redness, swelling, pain, and loss of function. What happens is when tissues become involved in inflammation, they stop doing their normal jobs in order to meet the inflammatory need. And so there is a loss of function there, anything from mobility to the pain causing, triggering you not wanting to use it, to abrogation of barrier function, like say in your digestive tract. If you have inflammation in your colon, you end up with diarrhea, so forth. So there's a lot to unpack in that idea of loss of function. Alongside the inflammatory side, you have cells that will, if they encounter potential pathogens and or cellular debris and so forth, they will phagocytose it. We call those phagocytes. All right, they wander through tissues, through the bloodstream, engulfing and removing foreign material. That can include dead cells or dying cells, pieces of cells, and also pathogens like bacteria or viral infected cells and so forth. These cells that are damaged and infected will release those cytokines we've been talking about, like IL-1 and so forth, and that triggers phagocyte migration to that area. That's known as chemotaxis. There's a whole family of cytokines called chemokines that really their primary role seems to be attracting phagocytes to an area of infection. The major phagocytes that we have, neutrophils, they're the most numerous of the white blood cells that we have, and they will actually get sticky to certain endothelial cells that are activated. So if there's an area of infection, the endothelial cells will actually begin to get sticky to neutrophils, and the neutrophils will kind of roll on the surface of the endothelium until they get to the area of infection, and then they will move out of the bloodstream. This is called marginalization and then diapedesis. This is just the cell can squeeze through the cracks of the capillaries, in an area where an infection is occurring. Then we also have macrophages. Macrophages are actually specialized monocytes. They develop into macrophages, and these often are found in tissues themselves. So you would find macrophages, like say in muscle tissue, or you'll find macrophages in the nervous tissue of the central nervous system, and so forth. We do have a link. There's a link here at the bottom. Recommend you have a look at it on YouTube. And so here we have a color enhanced picture of a phagocyte, probably a neutrophil, that is actively ingesting bacteria. Now, this phrase here, it says, if the first and second lines are not successful, well, yeah, I mean, it's all one large system. So it's a little bit prejudicial to say that, well, if we can't knock it out this way, then we have to go to adaptive. That's actually not true. This is a spider web. Everything is working together all the time. But what we are talking about here, the elements of what we often refer to as quote unquote immunity. First of all, there's memory. That's what we talk about. It's implied often when we say immunity that you've been exposed to it and now you have immunity. So the body does have cells that are now actively monitoring for a specific pathogen. It does recognize self versus non-self most of the time. There are situations of what we call autoimmunity, where tissues and the markers on the tissues then trigger an immune response that shouldn't occur. A classic example of this would be Graves' disease or Hashimoto's syndrome, which are autoimmune disorders on the thyroid. In any case, you're supposed to be able to recognize self versus non-self, and it should be specific. There are times, again, when it's non-specific, that can cause a lot of problems, but these are kind of the three major traits of what we refer to as adaptive immunity. The cells that are mostly tasked with this acquired immune response are known as lymphocytes, involves the lymphatic system. We have B lymphocytes. These are the ones that become the cells when they are triggered and differentiate. They can then create antibodies. We also have cells that then go out and can attack things directly. These are what we call the T lymphocytes, sometimes thymocyte. That's where the T comes from. Natural killer cells. These will attack virus-infected cells and tumors. And we also have these other ones called cytotoxic cells. Everything kind of coordinates. So they don't do anything by themselves. They actually create overlapping and redundant systems of immune defense. So T lymphocytes, these are the coordinators. In fact, a certain type of what we call helper T cell is what gets attacked by HIV. And so HIV depletes these helper T cells and the body becomes immunodeficient. That's what AIDS stands for, is acquired immunodeficiency syndrome. So T lymphocytes help mediate this cell-mediated. They provide signals that recruit and cause cells to recognize self versus non-self and whether we have an infection or not, so forth. They're called T because they are found in the thymus gland. We also have B lymphocytes. We've already talked about those. When they differentiate into what we call plasma cells, these are B lymphocytes that have been differentiated, and they are making a certain kind of antibody to a certain type of antigen. We call those antibodies a humoral. That's because they're diffusible. They're soluble in the blood, so they're in the humor of the blood, if you will. All right. These are memory cells. B cells become memory cells. They make antibodies, and they generally are in the once they mature, they're in the bone marrow. They might start in the lymphatic, but they do eventually move to the bone marrow where they are producing antibodies and so forth. Then you have macrophages and dendritic cells. These are phagocytes. They also serve as antigen presenting cells. They're called dendritic cells because they have these long finger-like projections, dendrites, like what we see in neurons. Now, this image is a little bit tricky to follow. It's showing you the process of how we make antibodies versus how we initiate cell-mediated attack, phagocytosis or killing of a cell. What's happening is that your body sees an antigen or an immunogen, sometimes it's called. And that will then turn cells on, which will then begin releasing signals down these pathways. If it's something that can be phagocytosed and then be presented to the helper T cell, which can then release signals to turn on cytotoxic cells, which then attack, kill, and phagocytose. On the other hand, you can have cells that are going to mature into antibody secreting cells. We call those plasma cells. And again, it's a similar process. You have some sort of an antigen that gets presented, and then a cell will then be activated to create a unique antibody to that antigen. And you can do multiple of these at a time. This is why you can receive multiple vaccines, because you have populations that are reactive to different antigens. And so you're not going to just swamp the system by giving them a few vaccines at the same time. A little bit about the T helper cell, also known as a CD4. That's the name of the molecule it's found that's on the surface of these cells. That's how we identify them. An antigen presenting cell, whether it's a dendritic cell or a macrophage or a neutrophil, after it's phagocytosed the molecule, will present it on its cell surface in a certain way that a CD4 cell, a T helper cell, will then interact with it. It's a physical contact, and it will then start releasing signals, IL-1, interleukin-1, interleukin-2, which will turn on antibody-producing cells. It will also send out signals that turn on and recruit macrophages to that area, and perhaps the CD4 cell itself will turn into a cytotoxic cell. It's expressing a slightly different molecule on its surface, so it'll attack cancer and virus cells this way. So this is why those T4 helper cells, if you knock them out, lead to a broad spectrum immunodeficiency. Since I've been in college, or even maybe a little before I was in college, cytokines have really come onto the scene in the last 30 or so years. Cytokines were this nebulous thing, and they kind of threw people off because up until the 70s or the 80s, most people believed that hormones were the diffusible signal that went through the body and caused cellular responses. And then and hormones are made by very specific cells. But then these molecules called cytokines can be released by any nucleated cell. Any cell that is alive and has a nucleus is capable of synthesizing and releasing cytokines. All right. They're diffusible and they act sort of like hormones. And so we have different kinds. Some attract immune cells, some trigger the growth and response of cells, and so forth. We've talked about a few of these things, interleukins and interferons and chemokines a little bit. But these are all examples of, again, they're hormone-like molecules that diffuse through the blood plasma and cause cell responses. But instead of it being a dedicated endocrine cell, it is any nucleated cell that is under duress will release cytokines. And cytokines are what drive inflammation. And now we're moving into the most specific type of immunity, and that is antibody-mediated immunity with this GAMED acronym. That stands for IgG, A, M, E, and D. Basically, from the most common to the least common. All right. Most common, IgG. All right. Most of the time, we see these late in a first-time infection, very late. But if we get that same pathogen infecting us again, these guys pop up really, really quickly. This is part of the memory side of the equation, and often IgGs will reside in your bloodstream for months or years after an infection. IgGs can cross the placental membrane, so they are transferred from mother to fetus. If you remember the plus minus in blood typing, the antibodies that will attack plus are what lead to the issue with we talked about ROGAM and a plus baby with a minus mother. That's one of the issues there. IgAs, we find these in our saliva, we find it in our tears, we find it in body secretions. It's a really interesting structure. I don't think we're gonna go into structure, but it's present when we release secretions in the body. We find it in vaginal secretions. We find it lining our mucous membranes and so forth. Before we have IgGs in an infection, when we the first time we're responding to a new infection that we've not seen before, the antibody that's released is called an IgM, and it's actually a pentamer. It's actually it's got 5 different heads on it. It's huge. All right. There may be some complement aspects to it, but in any case, what happens is these are released very, very first. And then following that, you get a class switch and you start releasing IgGs later on. IgEs are kind of funky. They show up during allergic reactions. All right. And then we finally have these IgDs, and they're a little bit unclear. We find them they maybe actually be receptors rather than true antibodies. And they do seem to help with binding antigens or something, so they're not the same as most of these other antibodies in terms of mounting a response, other than being helper, receptors on B cells. We do sometimes hear the term monoclonal antibody. What happens is that you can take a B cell that's making a given antibody, and you can isolate it in a lab and fuse it to make it immortal, and then it will just keep on releasing those antibodies. You can pure those antibodies to use for things like diagnostic tests. We can use it to give to people if they may be exposed to a potential pathogen, like we call a gamma globulin, that sort of thing. So they're very, very useful clinically and therapeutically. This image here is the basic functional structure of an antibody. Like I said, the IgM would have 5 of these, and the IgA would actually have 2 of them. But again, these are the functional areas of an antibody. You have up here, and this is basically the reactive area, the antigen binding sites, an area that's variable because we make antibodies specific to different types of antigens. And so in order to make it specific, you have to be able to modify the genetic code in order to make these regions bind specifically to the antigen in question. We have a light chain. This is the shorter one. There's another light chain over here. There's a covalent bond that links it to the longer one, the heavy chain. Both the light and the heavy have a variable region, and then they also have what's called a constant region. So these areas in purple do not change and are often involved in allowing, like for instance, phagocytes or other cells to recognize these antibodies down here, which means they can bind and do that sort of a thing. This is for binding the pathogen, and this is for perhaps interacting with cells in the body to mediate responses. And if one picture wasn't enough, it's just showing you this just in a different form. There's again the variable regions up here Constant regions down here, light chain, heavy chain, etc. Yet another picture, same thing. Can you recognize the antigen binding site? That's the variable region. The constant region is more for interactivity with the rest of the immune system. This photo here illustrates that idea of primary versus secondary infection and response. If you are exposed to an antigen, let's assume it's a pathogen, you will make a primary response. These are antibodies. Now, it's not saying what class. All right, at first, it's going to be primarily IgM. Now, if it's an allergic, no, I don't want to go there. All right, so primarily IgM, and then at the end, it will be IgG. And then time goes by, and maybe we get a second infection, and this time it's all IgG. But look how much more robust the response is. You might remember a few slides back, I said it's a numbers game. It's the number of players on offense versus the number of players on defense. Well, we just called the cavalry, and now we're bringing the whole store with us. All right. So this secondary response, much more robust, and will tamp down infection much more quickly. And this is the power of vaccines. What happens is you give a vaccine and you'll get a primary response, but then you'll have memory. So if you see the actual pathogen, now you're packing a punch. Maybe you see two pathogens at the same time. Now, here's one that you've had a primary exposure to. Well, this one's a novel pathogen. You hadn't seen it before, so you're getting IgMs and responses to that one that are similar to what we see over here. Because we haven't seen this before, we're getting a primary response, but because we have seen this one before, it's a secondary. So what do antibodies actually do? Well, for starters, they'll bind pathogens and prevent their ability to invade cells. We call this neutralization. That is, the antibodies kind of coat the pathogen, and so now the pathogens are just unable to do anything in the body. A similar response, this is more for IgM. That's an IgM pentamer there, so kind of a primary response. What you're doing is you're capturing what's called agglutinization. This works pretty well. There are some downsides to this, particularly if you're talking about sepsis. You can also precipitate, that is to say, make these things phagocytosable. And antibodies can also bind cells and trigger apoptosis in foreign cells or cells that have been targeted by the antibodies. So an example of neutralized or captured antibodies, we call this process opsonization. It's just an example of this right here that will then make those constant regions on the antibodies become flags for macrophages and neutrophils to phagocytose. So neutralization, capture, and precipitation are all triggering phagocytosis. Apoptosis can be triggered by antibodies, but they can also bind the cell, and then you get natural killer cells and so forth that will also work together to lead to cell destruction in one way or another. As if innate and adaptive immunity weren't enough, we do have subdivisions of adaptive immunity. All right. So adaptive immunity can be active. That is to say, you've actually been exposed to the antigen, either through a vaccine or direct pathogen exposure. All right. So you see the antigens, you develop antibodies and memory cells and so forth to them. That's called active. You have to be exposed to the antigen in the first place, either artificially through vaccines or accidentally, naturally through experience. Then you also have passive adaptive immunity. And this is when you get antibodies that you did not yourself make. So you can get gamma globulin in an injection, like for instance, if you're going to go to certain areas of the world that may have, for instance, certain types of diseases that you just don't want to get experience to, they can give you the antibodies ahead of time. And those will last for a period of time in your body and render you relatively immune to that potential pathogen. You can also inherit antibodies from your mother through the placenta and through secretion in the breast milk, IgAs in that situation. This is one of the reasons why, if you ever have a child, the worst thing you can possibly do is sequester that child in your house away from all the other people out there early on in that child's development. It would actually be better right after that child is born to go out to the grocery store and go out and into the world. Because when the child is born, they have a boatload of antibodies from the mother that are going to go away. So the best thing to do would be to have exposure while you've got all those antibodies. You're less likely to get very, very sick. But in our society, it is all too common for people to kind of sequester that kid for a few weeks, and then by then the antibodies have diminished, so that child's actually more likely to get sick. Okay, so it's actually better to get them out in the world. Remember that we evolved with babies being born out in the world. Okay, hospitals didn't come along with humankind, if you know what I mean. All right, never mind detergents and antiseptics and that sort of thing. These babies were born in rice paddies and in the savannah and so forth, exposed to antigens right from the get-go. So maybe we need to kind of return to that, because we are seeing increased instances of things like type 1 diabetes and autoimmune disorders and allergies that are associated with what they call the hygiene hypothesis. That is to say, the babies are in too clean of an environment early on and therefore are more susceptible. The last kind of passive immunity can be a monoclonal antibody. Again, it's sort of similar to gamma globulin. These things can be used in immunotherapy for certain treatments. Infections that are already occurring, like you can add these things to like, for instance, during COVID, there were certain notable patients who received monoclonal antibody therapy to help stave off their COVID infections, or diagnostics and so forth. And so there are four general ways when you have the active and the passive. There are sort of four ways that you can acquire immunity. One is the least desirable, the most dangerous. All right, that's called natural acquired active immunity. You actually have to get sick. There are benefits in some regards, but getting sick is a dangerous prospect. All right, it is very unpleasant. Why would we go through this if we don't have to, is the real question. All right, you can have artificial acquired active immunity. All right, so it is acquired, it is active because you've been exposed to the antigen, but you've been exposed by virtue of a vaccine, which cannot get you sick. All right. The third thing is what we call natural acquired passive. This is either through transplacental transfer or through breast milk. And finally, artificial acquired passive, which is where you're injecting the antibody into the bloodstream to confer temporary immunity with no memory, unfortunately. Now, cells are cells, and sometimes cells do good things, and sometimes they just don't work so well. And there are lots of different disorders in the immune system, and we don't have time to go through all of them. They're really, really interesting. That's more suited for a pathophysiology advanced situation. Though something that's very common we call allergies. This is a type of hypersensitivity reaction where you are particularly sensitive to a given antigen that may or may not be pathogenic. For instance, pollen from a ragweed is not pathogenic. Nevertheless, what happens is the body recognizes it as a potential pathogen, and now you have release of histamine, which is what is normally the driver of the symptoms of an allergy: runny nose, itchiness, and that sort of a thing. And you may also have antibodies that are also around as well that then trigger inflammatory responses. And the degree of severity is the degree of systemic response that occurs. If it's a localized thing, like for instance, I have a little bit of poison ivy on my skin right now. It is certainly a little bit itchy in that spot, but I'm not getting systemic types of responses. It's entirely local in that situation. The more systemic, the more potentially dangerous it is. You also have disorders where you lose components of the immune system. Either the barriers are not functioning properly, or you have loss of cell populations that mediate cell type responses. That's what AIDS is. It's an acquired immunodeficiency syndrome. All right. Not all AIDS patients have the same exact symptoms. It depends on which cell populations and how severe the depletion actually is. Then we have cells the immune cells attacking our cells. What would normally be considered self tissue is no longer, for whatever reason, is no longer identified as self. And so the body starts treating it as non-self. So we've been going for about 45 minutes now, so I am going to go ahead and pause for a moment, and we will conclude the video at this time, and we will pick up with part 2 here in the next video. Talk to you soon.